Now I am pleased to introduce our third speaker, Xiyang Wang, the senior researcher from the Viral Vector PD department of ProBio. Xiyang graduated with a master's degree from Wuhan University in 2022. After graduation, she came to ProBio and joined the clinical sample production department to engage in project production and delivery. During this period, she participated in ProBio's first 50-liter suspended LVV project and the first 200-liter AAV vector project. Later in April 2024, she transferred to the current China AAV PD department to engage in downstream process development and order delivery of AAV vector production. In this talk, Xiyang will be discussing the topic about AAV overview and solutions in cell and gene therapy. Now I will hand it over to Xiyang Wang.
Thank you, Jessica. Can you hear me and see my screen?
I can hear you, but can't see your slide yet.
And now?
Yeah,
Now you can.
It works now. Yeah.
Okay, let's begin. Hello, everyone. Thank you for joining us today. And I'm very delighted to be here to present about the topic of AAV overview and the solutions of ProBio in cell and gene therapy. So over the next few minutes, we'll have a discuss about the role of AAV as viral vectors, and most importantly, how ProBio is developing a comprehensive and scalable process to accelerate the development and commercialization of cell and gene therapy. So here we mainly talk about the 2 points, AAV overview and application, challenges and solutions in AAV process development. Let's begin with the topic of AAV overview and application. What is AAV? The full name of AAV is adeno-associated virus. It belongs to the genus dependent virus of the family Parvoviride. It's the simplest single-stranded DNA defective virus discovered so far, and requires a helper virus to participate in replication. The wild-type AAV genome encodes 2 open reading frames, the rep and the cap, which are located between the inverted terminal repeats. We call it RTR on both sides. The RTRs play a decisive role in viral replication and packaging. The cap gene encodes the viral capsid printing, and the rep gene is involved in viral replication and integration. Comparing with the wild-type AAV, the recombinant AAV consists of the same capsid sequence and structure. But from this figure, we can see the recombinant AAV has all AAV printing coding sequences deleted and therapeutic gene expression box added in genome. The only viral-derived sequences are the RTRs. From the discovery of AAV in 1960s to the first drug was approved in 2012, today the recombinant AAV has become the main platform for in vivo gene delivery. As a drug for gene delivery, the AAV delivers the target gene through the receptor recognition. This picture shows the AAV transduction pathway. First, the AAV particles will be recognized and bound by the cell surface receptor, and after internalized together, the AAV particle and the receptor will enter the cell to form the endosomes. And after escaping from the endosomes, we can see some particles will be degraded into peptides and lose the function of gene delivery. And other particles will enter the nucleus through the nuclear pores and after uncoating, the AAV genome will be released, and after the DNA replication, the genome will form the dimers or monomers to express the target protein by transcription and translation, so completing all the whole process of gene delivery. Based on its powerful gene delivery ability, the AAV performs very well in disease treatment. This slide shows all the AAV drugs currently on the market worldwide. We can have a look. We can have a look. From the dose, you can see the dose varies greatly, with a small dosage for local use and a large dosage for systemic use. And from the indications, we also can see the indications mainly focus on the ophthalmology, blood diseases, neurological diseases, and muscle diseases. We have used several minutes to introduce what AAV and recombinant AAV is and the application. Next, I'd like to move on to the next part, the challenges and solutions in AAV process development at ProBio. At ProBio, we produce AAV vector using the HEC293 cell line, and the transfection system is 3 plasmid system, including the RC plasmid for synthesizing the replication protein and the capsid protein, the helper plasmid for supporting the replication and packaging of the AAV genome, and the last plasmid is the transfer plasmid for carrying the transgene. Packaging and producing AAV vectors is relatively simple and easy, but we can see the chart. From the cell thawing to the last AAV drug product filling, there are many steps involved in AAV vector production and purification. So at ProBio, achieve high quality and high purification AAV vectors remains highly challenging. So at ProBio, AAV suspension process will mainly foster these 4 challenges. The first one is cell clumping, low density, and low viability. The second one is low titer. And the third challenge is how to effectively remove the AAV empty capsid to reduce the impurities. And the last one is how to maintain the stability of AAV drug product. So facing these challenges, we ProBio firstly have self-developed the suspension cell line, the Powers 293. This cell line has clear evidence of traceability and monoclonality, and is ProBio's patent. And from these 2 pictures, we can see the Powers 293 also has good cell dispersion and high transfection efficiency. With high density and high viability, from this data we can see the Powers 293 has shorter doubling time, and the shorter doubling time can improve the AAV production efficiency. And comparing with other cell line in terms of the yield, we can see the table. The Powers 293 is applicable to different serotypes of AAV. Having a good cell line also requires matching upstream and downstream process development. In the upstream process development, the main goals are increasing the yield and reducing the impurities. So the main optimization parameters are the media, transfection reagent, transfection parameters, harvest time, and so on. First, from these 2 figures, we can see in the media test, when selecting the media from the supplier B, the AAV titer can be increased by 3.6 times. And in the reagent test, we can see when selecting the transfection reagent from the supplier A, the AAV titer increased by 35%. Also, we have conducted a DOE study of transfection parameters and feed screening to optimize the transfection cell density, plasmid amount, and screen different feeds to increase the AV titer. We can see in this figure. The DOE model can predict the optimal transfection parameters to achieve the increased titer of 4.6 times. And in the feed screening, we can see using the feed A and B, the AV titer can be increased by 3 times. Let's see the harvest time optimization. When harvesting the AAV vector at 72 hours post-transfection, the AAV titer can be increased by 30%. So this harvest time is more appropriate for AAV. And there are two kinds of nucleases can be used to remove the host DNA residues and the residual plasmids. The salt-active and the non-salt-active nucleus. But from the data, we can see the salt-active nucleases remove the impurities more effectively. The last parameter of the upstream process is the different membranes and pore sizes in the step of clarification filtration. Optimizing this step can reduce the turbidity of the silicate and ensure the turbidity meeting the requirements of the downstream tangential flow filtration. From the figure, we can see after optimization, the turbidity of the cell lysate was reduced to less than 20 NTU, and the high recovery was also maintained. We have discussed about the upstream process development, and next we'll have a look about the downstream process development. The downstream process development mainly focuses on the two steps of chromatography. First one is affinity chromatography. We call it AC. And the second one is anion exchange chromatography. We call it AEX. Affinity chromatography can effectively separate the AV particles and impurities and remove the HCD and the plasmid DNA residue. In AC step, the key optimization parameters are wash buffer, loading capacity, retention time, and elution buffer. In the first wash buffer screening, you can see using the wash buffer tool can increase the recovery. It means the wash buffer tool is more appropriate for AC step. And in the binding capacity study, you can see with an increasement of the loading amount, the recovery was relatively increased. So it means that the loading amount cannot be too low for AC. And in the retention time, we can see using the time tool as retention time, the recovery increased. And from the last two figures, we can see elution buffer tool can increase the recovery and reduce the HCD and plasmid DNA residue more effectively. So it means that this elution buffer is more suitable for AC step. So in short, optimization of AC process can result in a higher recovery rate of 90% and can effectively remove impurities to meet the quality requirements. After getting the roughly purified AV vectors by AC, EX is also required to remove the AV empty capsid and further remove the impurities. In AX chromatography, the key optimization parameters are buffer screening, binding capacity, and elution strategy. In the first buffer screening, we can see from these two figures. Using buffer 2 and buffer 3 can increase the recovery and reduce the HCD, HCP, and plasmid residue more effectively. So we can see these two kinds of buffers is more appropriate for AX. And in the binding study, we can see with an increasement of the loading amount, the recovery was not affected, and the full capsid ratio is relatively increased. But if the loading amount was too high, the recovery decreased. So we can make a conclusion is that we can increase the loading amount with an allowed range to reduce the duration of AX and lower our costs. Let's see the last figure. In the elution strategy, we can see using strategy 1 and strategy 2, we can get a higher recovery. So in summary, AX process development can achieve high recovery rate of 60% and AV full capsid ratio greater than 70%. Also can effectively remove the impurities and meet quality requirements. After completing the upstream and downstream process development, the formulation research is also need to be conducted. Through the formulation buffer system and the excipient screening studies, the AAV can maintain stable titers and monomer properties to avoid aggregation. From these figures, we can see we have conducted many stability experiments on different buffers and different conditions, like the long-term condition, accelerated condition, high temperature condition, and so on. From the results, we can determine what formulation is more suitable for AAV storage and transport. So after the comprehensive process development, WeProBio has established a complete AAV suspension production process suitable for different serotypes. Under our process, we can achieve the titer of several serotypes greater than 2 times 10 to 14 Vg per liter. The total recovery rate of 30% to 50%, and the low impurity content and meeting the quality standards. Besides, from these AOC test results, we can also achieve the AV9-MD ratio less than 10% and the AV6-MD ratio less than 20%. Here we can show a projector case. Under our process, the projector is stably scaled up to 200 liters. These are the upstream data. From the figure, we can see after optimization, the AV titer after lysis increased by about 6 times, and the production process was stably scaled up from the demo run to 10 liters, 50 liters, to the last 200 liters. And the titer was stable at 5 times 10 to 13 Vg per liter. About the impurities, we can see after process development, the impurities are significantly reduced. And at the 200 liter scale, we can see the HCD residue was less than 5 nanogram per milliliter, and the plasmid DNA residue was less than 1.7% Vg. These are the downstream data. We can see from this figure, the downstream process remained robust from 2 liter to 200 liter. The recovery rate, the control of HCD and the plasmid DNA residue remained consistent. And also from the AOC test results, we can see the AAV empty ratio by AOC test was stable and was less than 18%. So from all this information, we can make a summary. ProBio has one-stop solutions. From plasmid to AAV vector, we have an overall solution to reduce the costs. And in the solution, we have built an innovative technology platform to accelerate the development. In the platform, we have our self-developed strain and our own AAV transfection system with the helper RC plasmid and self-developed suspension cell line, the Powers 293, and our own GMP MCB, WCB. Besides, we also have a high quality and stable process, and under the process, we can achieve high titer, stable recovery rate, low empty ratio, and stable scaling scaling of processes. And in addition, we also have a comprehensive and self-built DSDP quality analysis platform to ensure the quality control. Secondly, from the discovery to commercialization, ProBio can provide high-quality services at different phases, like from the plasmid design to in vivo pharmacology, from preclinical to BLA commercialization phases. So in short, ProBio has one-stop solutions from plasmid to AAV vector and from discovery to commercialization. The AV industry continues to develop, and as a participant, ProBio also face more challenges and difficulties. But ProBio will continue to seek more possibilities and to contribute the cell and gene therapy. That brings me to the end of my presentation today, and I'm very glad to be here to share our thoughts with all of you. Thank you for your time and attention. Thank you.
Thanks for Xiang's great sharing. Let's move to the Q&A part. Also, we warmly invite you to submit your questions in the chat panel and glad to invite you to follow us on LinkedIn and Kakao for more online resources and updates. Yeah, now let's take a look at the first question. Hi Xiang, when separating empty capsids, in which directions can optimization be performed? Could you please answer this question?
About this question is about the EX step. First, the choice of the elution buffer will affect the separation efficiency for empty and full capsid. For example, the potassium chloride is recommended in the Sim Malters QHR manual. It can be used. And we can also have a try about the sodium acetate and maybe have good results. Besides, the addition of the magnesium chloride will also promote the separation. It has mentioned in many papers. And the second, currently, we also commonly use the slow linear elution method. It can has a higher robustness, but we can also have a try about the gradient elution methods. They may can achieve the better baseline separation.
Yeah, thanks for your answer. And the next question is, I wonder every test you showed in your slides will be carried out for the process development of a new project. Could you support on this question? For a new project, we have for several for different serotypes of AV, we have
its own platform process. So if we have a new project, we will have a try using the platform process. If the result we is not really good, we will carry out the upstream and downstream process. Every test the upstream um, every test every test will be carried, and the downstream mainly focused on the AC and AX step. The TFF1 and the TFF2 we will took we will took much time. We will not took take not time much time about these two step, mainly focused on the chromatography.
Okay, thanks for your answer. And the next question is the origin of the proprietary cell line of ProBio is clear, or are there any cases of IND application?
About the cell line, yes, the cell line has clear traceability data, including the complete records of monoclonal screening and cell bank establishment, and has completed the DMF filling. The Powers 293, this cell line has successfully passed the IDA application for the AAV CMC project.
Thanks for the answer. And another question, I guess maybe Dr. Chen Luo, if you have any answer, you can also support on this question. How to avoid the liver toxicity?
Sure, thank you. Yeah, I and how to yeah you many recently clinical trials results demonstrate there might be some liver toxicity for AV drugs for AV candidates or drugs. I think mostly a quick answer. This can be solved in two ways. One is dose change or dose optimization. Efficacy or toxicology are always dependent. So if your candidate or our candidate is efficiency enough, we can give more potent candidates which can be dosed at relatively lower dosage. I think that will significantly reduce its toxicology. And the second, I think, is about the trypsin evolution. There might be ways, and we are constructing these capabilities for these, like optimization AAV capsid and to like impel its liver additive, and by I recently noticed there even might be some antibody retargeted decorated AAV to retargeted AAV to the and to other tissues like the target expression specific tissues to minimize its liver toxicity. Yeah, this is two ideas come up to my mind immediately. But I think after comprehensive literature review, there might be also some other ways to solve this. But this it's a really tough task for the AV drug development. Yeah.
Thanks for Dr. Luo's sharing. And another question, any speaker you have the answer, you can support on. Can you deliver to Germany? Yeah, I guess this is about the supply chain. Maybe Luffy or Xiang, do you have the answer of this question?
Uh, I think based for plasmid series, we have delivered some plasmid sample grade or GMP grade sample to Europe. I think it's okay.
Okay, thanks for your answer. Yeah. Also, we will ask our business development team to answer you more details through the email. Yeah.